Dear audience, my presentation is about studying functions of the Formins type of actin assembly factor in Drosophila melanogaster with the CRISPR system. Formins are conserved cytoskeleton regulatory proteins. They have essential roles in the actin nucleation, filament elongation, and bundling, and they also play a role in microtubule organization. Based on sequence comparison, formins are subdivided into 7 subfamilies, and our research focuses on the single Drosophila member of the DAAM subfamily. We got interested in this protein because it exhibits a number of interesting actin-related functions. Initially, we found that it is essential for the trachea development, and it is required also for the sarcomergenesis in the striated muscles. More recently, we are focusing on the central nervous system-related DAAM functions. We showed that it has essential role in the development of the neuromuscular junction by promoting the presynaptic Bushen formation. And it is also required for the development of the central nervous system by the axonal growth, promoting philopodia formation in the growth cone. We think that most of above activities are related to actin cytoskeleton regulation. But we also know that DAAM has a microtubule binding activity, and it can stabilize it. For example, it can protect the microtubules from cold-hitting or cold-induced depolymerization, as shown on these figures. So taken together, this formin is clearly an important protein with a number of interesting molecular function and tissue-specific function. But it remained partly unclear what is behind this complexity and whether isoforms play a role in that. Here you can see schematic structure of the Drosophila DAAM gene according to the flybase annotation. There are alternative transcripts with different transcription start sites and promoters. We thought to systematically examine each of these transcripts by creating isoform-specific mutations that were compared to the new mutation. This new mutant is lethal, and it has defects in central nervous system, neuromuscular junction, in trachea and muscle development. On the contrary, the RB specific isoform has function in the development of the adult central nervous system, and the RD is required for trachea and muscle development. My research is focusing on the RE and RA transcripts, for which we do not have specific mutants yet. Although they may affect interesting functions related to the embryonic central nervous system and neuromuscular junction development. My specific aim is to study the functions of RA and RE isoforms by creating isoform-specific deletions. To achieve this, I use the newly discovered CRISPR-Cas9 system, which is an ideal tool to create targeted mutagenesis. This system was first discovered in a bacteria where it has an antiviral function. The system is based on a protein-RNA complex. The protein component is Cas9, an RNA-associated endonuclease, and the RNA is short guide RNA, which has a designable 20 nucleotide long 5' end, which can recognize a specific DNA particle where the Cas9 cleaves the DNA. This target site must be followed by PEM sequence, an NGG sequence after the 3' end of target site. This is the protospacer adjacent motif. The workflow of my plan to create isoform-specific mutations was the following. First, I designed oligos for the CRISPR system, cleavage sites before and after the first exon and the transcription start. And after that, I cloned the oligos into appropriate plasmids and then inject these plasmid vectors into Drosophila embryos. In direct strategy, we inject these targeting constructs into Cas9 expressing strain. I carried out this research last year, but it was not successful, so I started it over with a modified strategy. We optimized the target site selection, and we used another plasmid vector. And instead of direct injection, we first create stable transformant lines. Our new targeting vector is PCEFD plasmid vector. This vector contains the regular ampicillin resistance gene, which is essential for the selection through the cloning. Moreover, it contains a wild type of vermilion gene, which is suitable for the selection after the cloning of the transformant animals. And it also contains an ATP site, which ensures the integration of the plasmid into genomic ATP site. This vector allows to clone 2 short guide RNA in one plasmid. And vermilion is a Drosophila eye color gene, which is suitable for the mutant animal selection through the crossings. We create stable transformant lines by injecting the targeting constructs into Drosophila embryos of indicated genotypes. Without going into details, around a few hundred embryos were injected, and I selected the transformant animals. Based on the vermilion gene, marker gene. Here you can see a figure of the integration, which is induced by the Phe-C31 integrals. And after selection, I made homozygous strains for each isoform. The integration was also confirmed by PCR, demonstrating that the targeting constructs landed into appropriate genomic region. To test whether our targeting constructs are able to generate the desired deletions, I've made a pilot experiment. During this, I crossed my homozygous strains to actin 5C Cas9 strain. This strain ensures the Cas9 expression in every cell. I made the PCR reaction with the progeny of this crossing. And besides the control products, shorter deletion products appears. The size of these shorter products corresponds exactly what expected if the deletion occurs. This pilot experiment is very promising and shows that our targeting constructs are able to generate targeted deletions. To achieve my final goal, I crossed my homozygous strains to Vasa Cas9 strain. This strain ensures the Cas9 expression only in the germline cells, so the mutation can be inherited into the next generation. Currently, I'm in the middle of this experiment. I will have to select for the deletion mutants and then establish stable stocks for each isoform-specific deletion. But based on my pilot experiment, I'm really close to the isolation and the phenotypic analysis of these isoform-specific mutations. I would like to thank to my supervisor Ede Mig and Mihály József and the developmental genetic groups of Biological Research Center. And thanks for your attention.